Okay, guys, it's going to be lab 3 about colony morphology. In theory, this is what we say. We say that a colony comes from a single bacteria and that we spread these out on the plate so that we can get the colony spread out because the shape and other characteristics of the colony help us with identification. Um, these are all basically what I've just got finished saying. The examination of plate cultures, acting, da 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 da. You know what? You guys can read that. It's really only telling you what we just said. That when we see these guys on our plates and we describe them, we can use those to look up the uh, the bacteria to which they could potentially belong, and then we use our biochemical test. So under here, you see that we have different forms. Now this is going to be the overall shape of the entire colony. Punctiform are a series of small little dots, circular, filamentous, irregular, rhizoid, and then spindle. So if you were to look at these guys on well, you don't need these for a microscope. These are visible to the eye. If you were to look at this, you should be able to classify this. We also, if you look at it from the side, the colony can either be flat, slightly raised, convex, pulvinate, umbinate. There's a little tip there on the top. And then the margin has to do with the actual edges more so than the overall. Those are 3 of the 5 things that we're going to classify these guys on. We'll talk about the rest here in just a minute. If we were doing this in the lab, you would actually receive these bacteria and that have already been grown, and you would use them to describe the colony morphology. Now, in addition to these 3, we can also talk about the color of the morphology. Is it white? Is it yellow? Is it, its appearance? Does it look kind of dull? Is it shiny? Does it look like mucoid, like it's wet? We would also be concerned with, can you see completely through it? Is it totally transparent? Is it partially translucent? You can barely see through it. Or is it totally opaque? Those are the kind of things that we'll use to describe colonies. The procedure they have, and absolutely we don't want to forget, we also in the lab we have little rulers that are marked off in millimeters. We actually measure from one side of the circle to the other, the diameter in millimeters. Pigmentation, the coloration, form, elevation, margin, and then the opacity. This is describing this Cynorhizobium meliloti. If you look there, they have decided that it has no pigmentation. It's clear, and I agree with that. I can see right through it. There does seem to be a little yellow dot there in the middle. I'd probably add that. They measured these from 1 to 2 millimeters. Opacity, they decided it is translucent for the most part. Texture, smooth, that's good. You could describe that as rough or whatever. The margin of the form they've said is circular, and that's pretty reasonable. The margin, the edges are entire. It looks like an unbroken, and I agree with that. And then something that we can sort of see, but we would probably need to look at it from the side. They've decided that elevation is convex. Guys, here are the videos. You really need to go and watch these. Colony morphology is just going to describe what I've just said, but it's good to hear it from a different person. And then this visual, I would definitely go there because they're going to show you different colony morphologies. Okay, here's your practice. They want you to take one of these three, look it up on the internet, look at and type in colony morphology, for example, E. coli or Enterococcus aeruginis, staph epidermidis, whatever. Take three of those, look them up on the internet, and then write out your description, and then you can check and make sure it's correct. Um, discussion questions. The first one is going to be really hard for you guys to answer because there really isn't. Well, I don't know. You could answer those based on doing these three. You certainly could. You could answer that question. What similarities did you see between the three organisms? And then you can also do number two. If the colony morphology is similar, how would you tell them apart? Which I already told you. You just use biochemical test. All right, guys, that's it for lab 3.